Hi everyone. This tutorial includes an overview of protein structure and talks about how you should think about this question, which focuses on protein denaturation. If you haven't read the question yet, pause the video now and take a minute to review it before we get started. Since the question asks about what levels of protein structure will be affected by denaturation, first, let's review how each level is defined. The first level of protein structure is primary structure. It is defined as the linear sequence of amino acids, starting from the N-terminus and going to the C-terminus. Here, we have a portion of the primary structure of the protein titin, which is important for muscle function. Each amino acid in this protein is indicated by its one-letter code name. It's important to know the primary structure of proteins, because every higher level of a protein structure is going to be dependent on its sequence. After primary structure, we have secondary structure. This is defined by two specific structural elements, alpha helices and beta sheets. The overall shapes of these structures are stabilized by hydrogen bonds between the atoms in the polypeptide backbone. While the amino acid side chains found in a portion of a protein will determine if that portion of the protein could form an alpha helix or a beta sheet, the interactions between the side chains are not defining characteristics of these structures. Rather, they are defined as having regular hydrogen bonds between amino and carbonyl groups from the polypeptide backbone. Both alpha helices and beta sheets are found in many different proteins, and each has specific structural characteristics. An alpha helix is made from a continuous stretch of amino acids that folds into a coil, with the side chain sticking out around it. Alpha helices are said to have a periodicity of approximately 3.7 amino acids, which means that it takes 3.7 amino acids to make one full turn in a helix. We can round this up to about 4 for simplicity when we are looking by eye at a protein sequence to determine if it is likely to form an alpha helix. Many different primary sequences have the ability to form an alpha helix, and the specific sequence of amino acids that make up an alpha helix can give you some idea of where that helix could be found in a protein. In class, we focused on alpha helices that are found on the surfaces of proteins. One way that this can happen is if one side or face of the helix is hydrophilic, able to interact with the water that surrounds the protein, while the other face is hydrophobic, so that it will bury itself on the inside of the protein. We can see this here if we look at this alpha helix as part of a protein that will be found in the aqueous environment of a cell. The paper cutout represents the overall shape of the protein, and the alpha helix is on the surface. The white pipe cleaner represents the polypeptide backbone, while colored pipe cleaners represent the side chains. As you can see here, the blue hydrophilic side chains face the aqueous environment of the cell, while the orange hydrophobic ones are buried on the interior of the protein. In this particular alpha helix, about half of the helix is buried on the inside of the protein, with the other half interacting with the water surrounding it. If we look at an unfolded version of this alpha helix, we can see how it was formed by looking at the characteristics of the amino acid side chains that make it up. The amino acids are paired up, 2 hydrophobic, then 2 hydrophilic, then 2 hydrophobic, etc. When the helix folded, these amino acids were positioned to make half of the helix, one face of it hydrophobic, and the other half of the helix hydrophilic. It's important to note that you don't necessarily have to have a half hydrophobic, half hydrophilic helix in order for one to be found on the surface of a protein. If more of the helix is sticking out into the water, maybe it's only a line of single amino acids that are hydrophobic, like we have here. This would also be enough to keep the helix stuck on the surface of the protein. In this case, you have 3 hydrophilic, then 1 hydrophobic, then 3 hydrophilic amino acids, etc. And you can also think about what you might expect to find if an alpha helix was entirely on the inside of a protein, or if a protein had an alpha helix that wasn't stuck to its surface, but instead extended out into the aqueous environment of the cell. There are lots of ways for alpha helices to form. The key things to keep in mind when deciding if a specific sequence would form a helix or not are, first, where the helix would be found in the protein and in the cell. Second, that the side chains stick out of the helix. And finally, that you start a new turn approximately every 4 amino acids. The other type of secondary structure is beta sheets. These are formed when extended stretches of amino acids line up, allowing hydrogen bonds to form between the atoms in the aligned portions of the polypeptide backbone. These stretches of amino acids can be from adjacent stretches of the primary sequence, or more distant ones, or even from 2 different polypeptides, as we saw with HIV protease. When the polypeptide backbone is in the fully extended conformation, the amino acids stick off alternating faces of the beta sheet. If one side of the beta sheet needs to be hydrophilic, and the other side needs to be hydrophobic, this occurs if the amino acid side chains alternate like this: hydrophobic, hydrophilic, hydrophobic, hydrophilic, etc. If both sides of the sheet should have the same chemical characteristics, then you would expect all of the amino acids to have the same properties, either being hydrophobic or hydrophilic, etc. Secondary structure focuses only on these local folds in the protein, individual alpha helices and beta sheets. When multiple secondary structural elements come together, and you add in the amino acids that aren't involved in these specific structures, but that connect them and contribute to the other folds in the protein, you're thinking about tertiary structure. Tertiary structure is the overall shape of a protein that's composed of a single polypeptide and is stabilized by interactions between amino acid side chains. Here we have another pipe cleaner protein, this time showing the tertiary structure. The polypeptide backbone this time is black, nonpolar side chains are orange, polar but uncharged side chains are green, positively charged side chains are blue, and negatively charged side chains are red. You can see that there are two alpha helices in this protein. They interact with each other due to the hydrophobic effect, which causes all of these orange nonpolar side chains to be buried on the inside of the protein. That interaction will also be stabilized by van der Waals forces. You can also see more specifically how which side chains you have in a sequence might influence whether or not it forms an alpha helix. Here, the helix is stabilized, as always, by hydrogen bonds between backbone atoms, but there is also the possibility for ionic interactions between positively and negatively charged side chains that are adjacent to each other in the helix, though not next to each other in the primary structure. This ionic bond here isn't itself a defining feature of alpha helices. They are defined by their hydrogen bonds, but it certainly would make it easier for this particular alpha helix to form. We also have this beta sheet down at the bottom here, which is formed from three so-called beta strands, two of which are adjacent to each other in the protein, and the third comes from the other end. Again, we have hydrophobic side chains facing the inside of the protein and hydrophilic side chains facing the outside. And we also have oppositely charged side chains that come together in the final folded protein to stabilize its overall shape by contributing to the formation of this beta sheet. And we have other folds that aren't part of any secondary structure, but which connect different parts of the protein. We can have ionic bonds or hydrogen bonds between side chains that help form these folds, or, if they were nonpolar amino acids, van der Waals forces and the hydrophobic effect. The last type of bond that is relevant for tertiary structure is covalent bonds between cysteines. Let's turn back to the question about BPTI to see why these bonds are important. In this question, we have essentially four key pieces of information to consider. First, most globular proteins are denatured and lose their activity when briefly heated to 65 degrees Celsius. Second, BPTI is a globular protein that has 58 amino acids in a single polypeptide chain, and BPTI has 3 disulfide bonds. Finally, unlike other proteins, after heating, BPTI is easily renatured. It folds back up into its functional form. We're asked to explain what levels of BPTI structure are disrupted by heating and why BPTI renatures so easily. So first, let's consider the first part of this question. Which levels of BPTI structure are disrupted by heating? We're looking at moderate heat here, so that's not going to affect covalent bonds. If covalent bonds remain intact, then primary structure is unaffected. What about secondary structure? Well, that's stabilized by hydrogen bonds. Hydrogen bonds are definitely disrupted by heat, so secondary structure would also be disrupted. In tertiary structure, as we just saw, tertiary structure depends on hydrogen bonds, ionic bonds, and van der Waals interactions, all of which can be disrupted by heat, so that is also disrupted. Disulfide bridges, though, are covalent, so while the overall shape of the protein will be disrupted, those specific bonds will remain intact. What about quaternary structure? Well, the types of bonds that stabilize quaternary structure are the same as those that stabilize tertiary structure, so quaternary structure would be disrupted by heat, except that BPTI doesn't have quaternary structure to disrupt. It's composed of 58 amino acids in a single polypeptide chain. Unless 2 polypeptides are involved, quaternary structure isn't present, so it can't be disrupted. Now let's take a look at the second part of the question. Why does BPTI renature so easily? And for this, I'm going to go back to my pipe cleaner demos. Here we have 2 very simple proteins representing BPTI, where we're focusing on the polypeptide backbone. There are no side chains indicated on this protein, and the only side chains shown on this one are the cysteines, which have formed disulfide bonds, which are covalent bonds, and so not disrupted by moderate heat. These proteins have the same overall fold in their polypeptide backbones. If I heat both of these proteins up, they will both unfold to the extent that they can. This protein, with no disulfide bonds, can unfold all the way. If it was allowed to cool, it would try to refold, but that would be very difficult, because the amino acids that need to come together in the properly folded protein are very far apart now, and will have a hard time finding each other again. In contrast, the 3 disulfide bonds in this protein will limit how unfolded it can get. The secondary and tertiary structures will become deformed and unfolded as much as they can, but the different parts of the protein are kept closer together, so that when the protein is allowed to cool, it's easier for the amino acids to find their binding partners again and refold into the functional protein. That is why BPTI renatures so easily. Voila!